celebrate the first Australians on whose traditional lands we meet and pay our respect to the elders, past and present. We also greatly appreciate the sponsorship of Lexogen, as well as Custom Science, the representative of Lexogens in Australia. Their portfolio includes kits for testing for COVID-19, characterizing small and messenger RNAs at single-cell level, and ribosomal RNA depletion kits. They can also help with engineering splicing controls for RNA-seq experiments. Please check out their products. I just put a link as well in the chat. It's a pleasure to meet you all online today. We send a warm welcome to our friends at the University of Canberra, Sydney, Melbourne, Adelaide, Perth, and all the attendees around Australia and worldwide. Today's presenter is Professor Manolis Kellis. Manolis Kellis is a professor of computer science at MIT, a member of the Broad Institute of MIT and Harvard, a principal investigator of the Computer Science and Artificial Intelligence Lab at MIT, and the head of MIT Computational Biology Group. His research includes disease circuitry, genetics, genomics, epigenomics, coding genes, non-coding RNAs, regulatory genomics, and computer genomics applied to Alzheimer's disease, obesity, schizophrenia, cardiac disorder, cancer, and many other disorders. He has led several large-scale genomics projects, including the Roadmap Epigenomic Project, the ENCODE project, the Genotype Tissue Expression Project, and Comparative Genomics Project in mammals, flies, and yeast. He received the US Presidential Early Career Award in Science and Engineering by US President Barack Obama, the Mendel Medal for Outstanding Achievement in Science, the NIH Director's Transformative Research Award, the Boston Patent Law Association Award, the NSF Career Award, the Alfred P. Sloan Fellowship, the Technology Review's TR41 recognition, the AIT Nicky Award, and the Sproul Award for the best PhD thesis in computer science at MIT. He's authored over 500 journal publications, cited more than 137,000 times. He has obtained more than 20 multi-year grants from the NIH, and his trainees hold faculty positions at Stanford, Harvard, CMU, McGill, Johns Hopkins, UCLA, and other top universities. He lived in Greece and France before moving to US, and he studied and conducted research at MIT, the Xerox Palo Alto Research Center, and the Cosmic Harbor Lab. These are all outstanding achievements. Please let me share some personal experience with his work as well. The ENCODE papers have helped grasp on how the eukaryotic genome and its regulatory regions are organized and determine cell-type-specific programs. This was one of the reasons I decided to stay in academia and do research on genomics. During my PhD work in Hungary, I characterized inflammation-sensitive enhancers in mouse macrophages. Manolis' method called RNA played a crucial role in this project. And finally, as an IT guy with a AI background, I often got uncertain whether life science is the right field for me. His exceptional career has demonstrated that such people can actually lead the field exactly because they're different perspectives and crucial skills. By this way, he also influences young aspiring scientists and make them believe that they can do the same one day. Therefore, Manolis Kellis is not only a great scientist, but a role model as well. Today, he will present one of his recent studies titled From Genomics to Therapeutics, Single-Cell Dissection and Manipulation of Disease Circuitry. We are happy to have Manolis Kellis here. Thank you very much.
Thank you so much for the very kind words, Attila. It's really very heartwarming to hear you. And I'm truly delighted to be here with so many of you. And thank you all for joining. What I'd love to tell you about today is our quest in translating genetic findings into therapeutic insights, and specifically not only how to dissect, but also how to manipulate the circuitry of human disease at single-cell resolution. So genetics goes back a long way. So basically for thousands of years, humans have selectively breeded crops, animals for their kindness, for their cognitive traits, for their food, for their wool. It wasn't until 1860 that Gregor Mendel published the first rules of genetic inheritance, the concept of discrete inheritance and no blending between different loci, the concept of dominant and recessive alleles and independent assortment of loci in different locations in the chromosomes. The challenge, however, was that Mendel's rules did not seem to apply to the seemingly continuous variation that we observed in human hair color, in human eyes, in human height, and so on and so forth. So a large school of biometrics basically pushed back against Mendel And his work was largely ignored for the next 50 years. Until Ronald Fisher, in the beginning of the 20th century, basically was able to show in very simple ways that even a small number, even 5 Mendelian loci that would contribute in additive ways can give rise to seemingly continuous distributions in height and other human traits. In fact, the slight deviations that could be seen from independent assortment in Mendel's experiments with bees were in fact used to map the distances between different loci. The further different genes were located in the chromosome, the more recombination, the more independent their assortment would be. And the closer they would be, the more correlation there would be between their loci. Mendelian trait mapping and dozens of genetic loci mapped early in the 1980s. But here we are today with complex traits becoming, for the first time, mapped systematically, understanding that not only Mendelian loci were seemingly additive models with thousands of contributing variables are now scattered across the human genome. We have more than 120,000 genetic loci that are robustly and statistically significantly associated with hundreds of human traits grouped in many groups here on this slide. So the challenge ahead of us is, how do we systematically understand the mechanism of these loci to truly gain insight into how to manipulate disease circuitry? Let me start with an example here in the FTO locus in chromosome 16. You basically have body mass index, a measure of obesity, showing the strongest genetic association in this locus. You also have dozens of additional associations, but this is the first genetic signal for obesity. Why is this exciting? This is exciting because genetics gives us causality. Genetics, in contrast to the correlational-based epidemiology that has been true throughout human history, genetics basically gives us a path to understanding the causal mechanism, to understanding that there's something going on in this region that causally impacts body mass index, and therefore can be manipulated in a causal fashion to combat the obesity epidemic in our civilization. So that's the promise of genetics, that basically by understanding these regions, we can understand the mechanism underlying disease. We can understand new target genes. We can develop new therapeutics and ultimately enable precision medicine and personalized medicine. So that's the promise. The challenge, however, is that when you look in this FTO locus, this fat and obesity associated locus, you basically see 89 common variants that are all co-inherited. We don't actually know which is the driver one simply by looking at this plot. Any of them could be responsible for the observed genetic association between these two recombination hotspots. So basically this entire region is co-inherited between different individuals. So that's the first challenge. The second challenge, by far the largest challenge, is that in 93% of cases, these disease hits are in fact not altering the protein coding region for these genes. In this particular case, you see this FTO gene has another exon here, a third exon here, and has these very large introns where the protein coding sequence is encoded only within the exons. So that basically means that these 89 common variants do not perturb the protein directly. And that also means that the target gene might just not be FTO. I was introduced as the author of one of the most depressing papers of 2015 when it was realized that even in those examples where there's a single gene underlying the genetic association, we couldn't be confident that that would be the correct gene. Basically, people are all often very disappointed when there's like 5 or 6 genes in a genetic locus. But in this particular case, it was a single one. And what we showed, in fact, in our work was that this was not even the correct one. So in our paper with Melina Klaas and certain collaborators, we basically showed that the true target genes are sitting 1.2 million nucleotides away. and 600,000 nucleotides away from these genetic locus. So the challenge of understanding mechanism using these genetic studies is that, number one, we don't know the causal variant. Number two, we don't even know the target gene. And that's not a weird example with FTO. In 93% of cases, these genetic loci do not contain protein altering variants. So it's not just an academic exercise. This is the vast majority of these complex traits. Are a new, fundamentally new way of understanding disease genetics, to find the target gene, to find the causal variant, and then to understand what cell type do they even act in. For obesity, is it governing our appetite, whether we eat, sweets, or whether we eat, care to exercise? Does it govern our muscles, and so on and so forth? So even understanding where these genetic variants are acting is a great challenge. And of course, if you don't know any of these, it's very difficult to understand the relevant pathways and the mechanisms. So the approach that we in my lab and many others in the field have taken to dissect the mechanism of genetic loci is to start with human genetics to reveal both common and rare variants and regions of genetic association. Here's another example for the ApoE locus underlying Alzheimer's disease and many other Alzheimer's associated genetic loci. To start with these genetics and then to systematically profile the transcriptome, so RNA, and epigenome, the circuitry governing RNA production in both healthy and disease samples. Healthy, to understand how genetic variants associated with disease are acting in isolation of the disease etiology. And we want to understand both the impact of genetic variants as well as the impact of environmental and other contributors to disease. So once we have generated, in my own lab, despite running a primarily computational lab, we have generated thousands of these data sets from understanding this transcriptional and epigenomic variation. We then integrate these data sets computationally to predict driver genes, regions, and cell types, to understand those circuits, and to make hypotheses about how we can manipulate the disease phenotypes. And then using these, we then go into cell cultures and mouse models to validate our predictions and to show that indeed this causality that we are inferring from genetics is indeed holding in interventional models. And then, of course, disseminate the results and start the cycle all over again. So here's an illustration of what this gives us. Instead of having just a region of association, what we have is individual genetic variants underlie this association. Basically, we're not just blind to the genetic variants, we know exactly what are the nucleotide substitutions. That allows us to now start looking for the regulatory control regions known as enhancers, these distal acting regulatory regions, where the genetic variants are sitting. And of course, the cell types where the genetic variants are acting by systematic epigenomic mapping, as I'm going to show you in a second. We then take the upstream regulators and their regulatory motifs to see which ones of those are actually disrupted by the SNPs and which ones are binding these regions. And lastly, what are the target genes? So basically that gives us the complete circuitry, giving us a transformation from just a region of association to a circuit and potentially causal mechanisms. In the case of FTO, basically this FTO obesity variant, we basically dissected its circuitry in the case of, and we looked at these 89 common variants. We were able to fine map them into a single genetic locus, into a single SNP, into a single genetic difference, a single letter that was in fact disrupting an evolutionarily conserved AT-rich motif, which in the obesity risk individual changes from a T to a C, disrupting the binding for this AT-rich interacting domain protein ARID5B. ARID5B normally binds this 12,000 nucleotide super enhancer, which is active in mesenchymal stem cells that ultimately give rise to either white fat or brown fat. And what we showed is that ARID5B is in fact normally repressing this enhancer, which then becomes de-repressed, no longer be bound, the motif disappears, and therefore this repressor of IRX3 and RX5 no longer represses them, they become de-repressed. And what we showed is that IRX3 and RX5 are two previously unknown master regulators of a process known as thermogenesis or heat generation. So thermogenesis is a process through which, through mitochondrial membrane depolarization, you can basically lose energy in the form of heat through a process known as browning or beiging of adipocytes. Thermogenesis is shut down. The only path for all of the excess calories in our diet is lipogenesis. So basically storing of that energy in these high energy molecules called lipids, which basically accumulate in white adipocytes and make them large. So it's not your appetite that is at the basis of this association. It's not your exercise, it's not your muscles, but it instead appears to be a master switch, a master metabolic switch inside your fat storing tissue, your fat, that basically decides between fat storing or a fat burning. Don't cover the circuitry, not just to tell people you have a mutation, but to actually intervene, to basically say that for those individuals where exercise and diet interventions appear to not be working, perhaps we can intervene directly and start manipulating their metabolism. So we can now start interventions. We can now start thinking about either increasing or decreasing the upstream regulator or the downstream target genes, IRX3 and IRX5, to shift between lean and obese phenotypes. And we can even use CRISPR-Cas9 genome editing in somatic interventions in adipocytes to basically switch from the, which disrupts the motif, back into a T, which restores the repression and therefore should restore a lean phenotype. Indeed, each of these interventions, as we show in our paper, has led to a dramatic shift between lean and obese phenotypes. What we basically showed is that in risk individuals, in primary adipocytes from risk individuals, we can basically shift from thermogenic to a thermogenic state, both in the control and in related conditions. So my own cells are basically homozygous risk. I am a double risk carrier for these obesity locus. Both my mom and my dad gave me these obesity risk alleles. And basically taking primary adipocytes from these double homozygous carriers, we basically show even more if we target genes, for example, IRX3, which gets de-repressed in obesity. We basically towards the lean phenotypes. And indeed, using adipose expressed IRX3 dominant negative construct, we basically show that mice lose about 50% of their body weight to start with. When they are put in a high-fat diet, normal mice gain weight, but these mice are unable to gain weight. So basically, they, if you open up their bodies, you basically see that their stores are completely depleted. All of their fat basically looks red. It's extremely thermogenic. And this is preventing them from would like to do systematically across the 120,000 genetic loci. We'd like to systematically understand the circuits in order to enable manipulation, reversal of the disease circuits. So how do we do that? We basically employ many methods. The first is comparative genomics. We need to understand even the basic foundations of every single nucleotide in the human genome. What are the functional regions of the human genome? Where are the motifs? Where are the protein coding genes? Where are the regulators? Where are they binding? So one such lens that we're using on the genome is the comparative genomics lens of evolutionary signatures. So we've developed a series of signatures through a series of papers, starting with my PhD and then my first postdoc, and several additional papers, where we have basically showed that if we look at how things evolve, if we look at evolutionary patterns of change, not the conserved nucleotides that I'm showing here in white, but if we look at the change, the altered nucleotides that I'm showing here in color, we see that protein coding regions evolve in a completely different way from non-coding regions, enabling us to distinguish them simply on the basis of their evolutionary signatures, using codon substitution frequencies, reading frame conservation, and basically build a reference human gene set. So my group has been collaborating with a GenCode consortium for almost 2 decades now, building the reference set of the human genome. We've also collaborated with many other scientists to build a reference set of RNA structures, of microRNA annotations, of regulatory motif annotations, of individual regulatory motif instances, each using their evolutionary signatures. And for every one of these slides, you can click on this. So in a recent paper that we basically just published a couple of years ago, we basically looked at 118 previously non-coding genome-wide association study hits. And what we basically showed is that they're in fact protein coding after all. Basically, we discovered many new protein coding exons in the human genome. In this particular case, there's a myopia-associated genetic variant, and there's exon here that we predicted, which is in fact retina specific. So we were simply blind to the fact that this region of association is in fact protein coding. Is the case for many additional traits, for 118 such hits that we can now show are coding. But much more importantly, we can basically go through the genome and detect these evolutionarily conserved nucleotide motif whose disruption can basically point to potentially causal mechanisms of disease. So here you have this SNP that disrupts an evolutionarily conserved G with an A, hint as to what might be happening. These annotations are very helpful for the human gene set, but even for SARS-CoV-2, the virus underlying the COVID-19 pandemic, which is only 29,000 nucleotides long, it still has many hypothetical ORFs. So using this genomics approach, we basically show that, for example, ORF10, which is one of the very few annotated ORFs for this, is in fact not a protein coding gene at all. It's in fact a non-coding RNA that accumulates mutations exactly as RNAs do. We were able to show that there's a new overlapping gene, ORF3C, that basically perfectly overlaps this ORF3A gene in a different frame. We were able to also study how these genes are evolving across many sarbecovirus viruses or within the current pandemic and find cases of acceleration. Also study the strains in a new this virus. So that's the first challenge. Basically, how do we build these reference comparative genomic maps to predict disease-relevant nucleotides? But the second tool that we're using, in addition to comparative genomics, is epigenomics. So this is looking at the epigenome, looking at the packaging of the DNA around nucleosomes and into chromatin. That basically gives us not just structural information, but also functional information, because DNA accessibility can greatly impact how different regulators can bind these regulators, turn genes on and off. And also the nucleosomes themselves, made of 8 histone proteins each, can themselves be greatly modified using post-translational modifications in the tails of these histone proteins that allow us to now start reading the signals that the chromatin itself is reading to, quote unquote, remember the state of different cells. So as you know, all of our cells in our body have essentially the same DNA, but they all have dramatically different functions. And based on whether this region is useful in a liver cell versus a brain cell, this region will be marked by different histone modifications. We can use the histone modifications themselves to understand what regions are turning on and turning off in different cell types. So as part of the epigenome roadmap project, we basically looked at a large number of these primary tissues from different regions of the body, different regions of the brain, different stages of development, and also different stages of differentiation for iPSCs, embryonic stem cells, and many in vitro differentiated or primary human cells, which I'm going to be color coding here. Associated, enhancer associated, transcription associated, polycomb repressed, heterochromatin, and simply activity, whether it's in enhancer or promoter regions. We also looked at DNA, a mark typically associated with repression. DNA accessibility is associated with TF binding, usually found for both active but also for repressed regions. And then, of course, the output, quote unquote, of gene expression. HMM for chromatin hidden Markov model that, quote unquote, learns the language of the epigenome de novo by scanning across the genome for dozens of these modifications, and then using an expectation maximization machine learning approach known as Baum-Welch for de novo learning. We basically built this multivariate hidden Markov model that allows us to learn the vector combinations of these modifications across the genome, and from that, learn that in fact, about 1% of the genome that was associated with this class, another 1% associated with that class, another 30% associated with this class, which we then gave names to according to the enrichments that they show for different classes of regions. So we have a signature for enhancers, for promoters, for transcribed, for repressed, for chromatic regions. And we can now use these to systematically annotate the epigenome of dozens of tissues. And what we can do is take these annotations and start overlapping them on the y-axis with genetic variants in the rows here that are associated with different traits. So for example, if we take genetic variants associated with height and we ask, where are they the most enriched across the enhancers that are active in different tissues, we see that they're enriched in embryonic stem cells, suggesting that genetic variants associated with height have probably a developmental role. If we look at genetic variants associated with diverse immune traits, they're enriched in enhancers active in T cells and B cells. Genetic variants associated with heart rate polarization, specifically enriched in the left ventricle of the heart. Genetic variants associated with cholesterol are enriched in liver enhancers. Genetic variants associated with fasting glucose-related traits and a signature of type 2 diabetes are very specifically associated with enhancers, enriched in enhancers associated with active in pancreatic islets. If you look at inflammatory enhancers and bowel enhancers in digestive tissues. If you look at Alzheimer's, that's where it gets a little surprising. We basically see that there's zero enrichments for enhancers active in brain. And that's, if you look at brain tissue, you basically have 90% of cells made out of neurons, oligodendrocytes, and astrocytes. Only 5 to 10% of cells are made out of microglia, which are in fact showing the CD14 plus marker, also found in circulating monocytes and tissue-resident macrophages in a diversity of tissues. So what we basically postulated is that perhaps the genetic variants associated with Alzheimer's are not acting through neurons, oligodendrocytes, and astrocytes primarily acting through microglia. So we carried out suspended single cells, and we put them through a flow sorter, selecting for markers of neurons, immune cells, and the rest. And what we basically found is that neuronal cells, the brain, showed no enrichment. Oligodendrocytes, astrocytes, and the rest of the cells showed no enrichment. The only enrichment was found very specifically for microglia using LD score regression for Alzheimer's associated variants, suggesting in fact that the mechanism associated with Alzheimer's might be primarily acting is involving microglia. Of course, they might also act in other cell types, but to a much lower degree. And we published a paper back in 2015, basically saying that in fact, conserved epigenomic signals in both mice and humans could reveal an immune. Collaborating with Li Wei-Tsai from the Picower Institute, the director of the Picower Institute, we basically looked at her model, PKP25 mice that develop both early, late stage symptoms for Alzheimer's. And what we basically showed is that early in time, we basically see only immune over repression, suggesting in fact that immune processes and inflammation might be a causal component of only smaller contributions on the genetic level. We have since expanded from 127 epigenomes to 834 epigenomes, from 54 enriched traits to hundreds of enriched traits. You can see here some of that enrichment matrix that I showed you earlier, but now we have hundreds of genetic traits on the rows and then 800 tissues grouped here on the columns. And what that basically gives us is a very rich interaction network between different phenotypes and on the names here, different traits and different tissues in the pie charts that I'm showing here. Human intelligence and so on and so forth, you basically see enrichments very specifically for enhancers that are active in primarily neurons, i.e. the brain tissues. If you look by contrast at Alzheimer's, you see that it instead localizes here in the immune traits. If you look at cholesterol, you see liver. If you look at filtering functions, you see kidney. If you look at coronary artery disease, you see heart, but you also see it localized here in the middle, in between all of these different tissues. So you can see these pie charts are much richer compared to those pie charts with, for example, only brain or only immune. And this is basically suggesting that some traits, such as coronary artery disease, are in fact very polyfactorial. There are many factors that are contributing to them. So if we take a look at coronary artery disease and we compare it to QT interval, educational attainment, Alzheimer's, BMI, and health span, you basically see a range of complexity. QT interval, very specifically heart tissues. Educational attainment, very specifically brain and early neurons. If you look at Alzheimer's, you see immune cells, but also brain with a lower degree. Look at waist-hip ratio, you start seeing more tissues, adipose, muscle, kidney. If you look at health span, you start seeing more again. And if you look at coronary artery disease, you see the most rich set of tissues that are enriched. So how do we deal with that? What we can basically do is use the epigenome to start partitioning out this complexity into smaller components. We can look at just the subset of genetic variants that are associated with that are overlapping liver enhancers, or the subset that overlap coronary artery enhancers, or the subset that overlap thyroid or adipose. And what we see is that when you only focus on that subset of variants, then the biological enrichments for gene function that you get are dramatically different between liver overlapping enhancers and coronary artery enhancers. You also see the same partitioning applying to the co-associations of comorbid traits, which differ between different subsets of enhancers. So we can now go down the list of genetic associations and basically see for coronary artery disease, what are the top enriched tissues and what are the enhancers that overlap there, and where are they seemingly acting? You can see here the distances between the genetic variants and the nearest enhancer. In this particular case, for PCSK9, you see there within 900 bases of a liver acting enhancer. And you can see here a plot. We basically constructed 30,000 of these plots in this URL here, compbio.mit.edu/epimap. where you can basically see the genetic associations and the linking between these genetic variants and nearby genes that we predict are in fact targets of these enhancers. You can see here a different example for the NRA. So number 2 here, you see it's within 60 nucleotides of a coronary artery active enhancer. You see number 3 here for this gene there, you basically see both. You see liver and also you see heart associated links, suggesting that perhaps this genetic locus is impacting perhaps through different genes, both liver functions and coronary artery functions. And in fact, this is not an exception. This is very common, suggesting that perhaps one of the reasons why some of these genetic loci have escaped selection is because of their pleiotropy. Basically, it's only when both of them overlap together that you end up having a phenotype appear in, for example. So using maps and these reference epigenomes to predict disease-relevant tissues through comparative genomics and these epigenomic maps of non-coding elements, what we can do now is start looking at how these maps vary between individuals, because the epigenome of each person is different and the genome of each person is different. So we'd like to understand how is the genetic their transcriptome and how both of these vary in the context of what we'd like to understand is how intermediate phenotypes mediate impact of genetic variants on disease. It's a very long path from genetics all the way to disease. You have tens of thousands of genetic loci associated with extremely weak contributions. But if you start bridging that path, you have much stronger effects at the enhancer level or at the gene expression level. And similarly, gene expression correlates more strongly with these endophenotypes because the path is much smaller, so you end up having stronger, more oligogenicity rather than polygenicity. The challenge, however, is that genetic variants are in fact bidirectional for these intermediate variables. And the reason that the disease might be in fact responsible for the genetic. In fact, the gene expression changes might be combating the disease. Men at the fire, they're not the arsonists. They're there because they're combating the disease. So this gene goes up for individuals with the disease, but reducing, make the disease even stronger rather than the other way around. So we really need to understand the path of causality to understand where we can intervene and in which direction we should be intervening and how it is impacting disease. We utilize the genome and utilize genetics to basically understand how the genetics impacts both the epigenome and the transcriptome through quantitative trait loci, either eQTLs, expression QTLs, or methylation QTLs. Number 2, how the methylome or the transcriptome correlate with phenotype through methylome-wide association studies. And of course, number 3, combining the two to understand causality. So looking at the first arrow here between genetic variants and the epigenome, we basically found 50,000 genetic loci that impact in post-mortem human brain at 90+ years of age. And here we're looking at the number of alleles carried. And what you can see is that the more alternate alleles we have, the higher. It predicts the methylome of a person 83 years of age. So basically, I can predict it decades earlier and quite reliably so for tens of thousands of these genetic variants. That now start doing all kinds of really cool things with it. We're looking at GWAS, this very large arrow that I mentioned earlier, or QTLs, which are basically looking at this molecular intermediate phenotype, or this correlation, this bidirectional arrow of methylome-wide association studies. I can first use the QTLs to predict, to impute methylation. And that gives me a unidirectional arrow. And this is not all of methylation. This is the genetic component of methylation. I can use that to start predicting the disease state. And if I use that, that basically means that I get my unidirectional arrow back, which gives me causality. I get the relevant tissue where the genetic variants are acting. I get the intermediate molecular signal localized to specific enhancer. So that gives me, again, the target. And if I can do that with both methylation and, for example, transcription, it also gives me intermediate genes that might be mediating these effects. And it also increases my power because I can apply that to tens of thousands of individuals. All I need is their genetics because I have trained my model on a few hundred individuals. And therefore, because I'm looking at common variants, to tens of thousands of individuals that carry these common variants. So just basically look at locations, for example, here, all of chromosome 6, which has no genome-wide significant hits for Alzheimer's, and then start predicting a large number of novel hits because I'm combining multiple SNPs together to start predicting. And doing this through multiple intermediate variables basically allows me to start predicting driver genes in locations of genome-wide significance, as I'm showing here in purple, their directionality of effect, whether increased or decreased in the case of disease, and also new loci that are now in these gray loci that are not genome-wide significant. Showed you so far has been at the tissue level. What we would like to do is actually start looking at the single cell level. So in my lab, we basically looked at a large number of diseases in multiple collaborations with Wed9am, with David Bennett, with Rudy Tanzi, with Veronique Belzil, with many collaborators, looking across Alzheimer's, frontotemporal dementia, Lewy body dementia, ALS, Huntington's disease with Miriam Heyman, AD and psychosis with Robert Sweet and Yulia Koffler, schizophrenia with Brad Ruzicka, bipolar disorder, Down syndrome with Elizabeth, depression, and so on and so forth. So for each of those, we basically have dozens of individuals for whom we can basically look at neuron-specific, oligodendrocyte-specific, astrocyte-specific, microglia-specific variation. And we can also look at different brain regions and look at both transcriptional and epigenomic changes. What we've seen is that for Alzheimer's disease, in the first paper that we published a couple of years ago, we basically looked at non-AD, early AD, and late AD individuals. And we basically started distinguishing the effect across different cell types. We basically showed that even though genetic variants were primarily enriched in microglia, we found transcriptional alterations in nearly all of the cell types. And these effects were very strong, specifically for female individuals, and basically showed an abundance of these disease-associated transcriptional states. We found dramatic differences between early increase in expression in Alzheimer's, which was very cell type specific, and late increase in expression, which was very conserved across different cell types, suggesting perhaps a common response to the damage that happens with neurodegeneration. We found dramatic differences between male and female brains even before AD, but these differences were exacerbated in AD. We have 3,000 differentially expressed genes between males and females in excitatory neurons, and that turns into 6,000 for Alzheimer's. This points to perhaps sex-specific interventional treatments where women, for example, showed much greater white matter loss than men, associated also with differences in oligodendrocyte expression, which of course, we have since expanded from simply the prefrontal cortex to 7 different brain regions. We've expanded the number of individuals as well. We now have 1.6 million cells across inhibitory neurons. We have now 30 different excitatory, 23 inhibitory neuron types. We basically see great variation between different brain regions for inhibitory neurons, as well as for glial cells. You can see here that astrocytes vary dramatically between different brain regions. You can see here thalamus versus mammillary body astrocytes are dramatically different, as well as subcortical versus cortical regions. We can also use all of these cells to start sorting cells according to their transcriptional signatures of Alzheimer's disease. And we can distinguish genes that are differentially expressed in Alzheimer's early or late in the disease process to start thinking about early interventions. Look at the signatures of these cells, and what we found is that the clusters that we're discovering are in fact reflecting anatomical subtypes of neurons that are also showing dramatic differences in morphology, enabling us to predict, for example, that CA1 neurons in the hippocampal formation here are in fact much more strongly associated with Alzheimer's disease than dentate gyrus neurons. In realization, we basically started deconvolving the spatial and transcriptional changes that are happening, in this particular in the cerebellum with ACA1, a spinal ataxia in the cerebellum that is associated with movement disorders. And we basically show that Purkinje cells are in fact dramatically reduced when you look at the deconvolution, even though their markers are not noticeably different. And you can see the spatial organization is actually very often lost in SCA1. We also developed a deep learning model to start the coordinates of these cells by training it on these sort of combined transcriptional and spatial data. Basically learn modules of genes that are co-varying across these millions of cells to basically start understanding these as a group, which gives us a much more robust signal than single gene inferences. And we can see how these modules are in fact dramatically altered in different cell types. Focusing just on microglia, we can basically see phagosome versus glycolysis versus chaperone versus DNA damage modules differentially associated with different phenotypic subtypes of Alzheimer's disease. Also look at the correlation of these modules between different brain regions. And for example, see that the neurons that are connected through known circuits between different layers of the entorhinal cortex versus the hippocampal formation, in fact, correlated in their changes in Alzheimer's between individuals, enabling us to in fact start inferring that perhaps there's something about the connectivity that helps either the loss of activity that might be leading to neurodegeneration. I mentioned we've done this across many disorders, looking at schizophrenia, for example. We looked at these hundreds of genetic hits associated with schizophrenia. And for each of those, we were able to see what are the differentially expressed genes in this locus, which are very often supported by high-C three-dimensional conformation capture of the chromatin evidence linking genetic variants to these genes, and also what cell types appear to be the most dysregulated for each of those genes. We're able to predict schizophrenia versus control individuals from their transcriptional signatures. In some cases, predicting that this control here shows schizophrenia-like patterns. And even though that individual was not diagnosed with schizophrenia, his son was in fact diagnosed, suggesting that perhaps some of these transcriptional alterations might have predisposed the father, even though the disease only manifested in the son. We also see control-like patterns for a subset of schizophrenia cases, and these are in fact enriched for this SDTR cell state, suggesting perhaps either a separate path of transcriptional resilience. Using both early in development as well as in adulthood. In the case of psychosis, in the context of Alzheimer's, in contrast to schizophrenia, there's Alzheimer's-induced psychosis. And this is associated with a lot of repression of not overstimulation that leads to psychosis, it's instead activity in the neurons And we can see several processes associated with this: ribosome, neuronal. ALS and FTLD with Veronique Belzile and Miriam Heyman in work led by Sebastian Pineda, we basically showed that, in fact, even though the two diseases are quite distinct in their phenotype, one is affecting motor, the other one is affecting cognition and behavior. We showed, in fact, is that the same cell types in the motor cortex versus the frontal cortex, it's a typo here, are in fact affecting the same long-range projecting layer 5 excitatory neurons. So these basically suggest that perhaps the two diseases are orthologous, not only in their genetic underpinning, where you can see this C9orf72 association with both ALS and FTD, the specific genes that are the most dysregulated. In the context of Huntington's disease, again, in work joined with Miriam Heyman, we basically show that indirect pathway spanning projection neurons and direct pathway spanning projection neurons are dramatically distinct transcription. In the context of pathogen-associated changes in AD, in collaboration with Rudy Tanzi, we basically looked at cytomegalovirus and human simplex virus 1 and their association with disease. There's a major hypothesis of an immune basis of neurodegenerative disorder, where amyloid beta oligomers would be serving as traps for viruses and pathogens and would be increasing their presence in the context of infection, either leading to amyloid beta accumulation and possibly looking at HSV1 versus non-HSV1 individuals, we basically showed that indeed there are dramatic pathway differences associated with immune function in inflammation, in synaptic transmission, and other impacts of viral infection. We also use these data to start understanding the effect of strong effect variants associated with ApoE, ABCA pathways, which are basically having a strong transcriptional impact on disease. We are able to distinguish different subtypes of microglia having such a large number of cells, and then how, in fact, homeostatic microglia do not appear to be a naive default state, but in fact have to be actively maintained. And these are, in fact, the ones that are showing the strongest expression alterations in the case of Alzheimer's disease. We recently published a paper in vasculature dissection of both ex vivo using epileptic samples of living human brains as well as post-mortem brains from our collection to basically look at the definitions of different vascular cell types. These constitute only 0.3% of cells of the brain, able to infer their transcriptional gradient between these venule versus arterial zonation of endothelial cells, pericytes, and smooth muscle cells. And we're able to distinguish a new subtype of fibroblast that appears to be a human-specific and mainly disease-associated changes in the vasculature. In a paper that we just posted in bioRxiv, we basically see that many of the genetic variants associated with Alzheimer's are in fact showing differential expression in the vasculature with very strong enrichments, and again, support by linking through EpiMap, through GTEx QTLs, single-cell EQTLs, and high-C data, leading to a lot of related pathways, and possibly acting through interactions between these vascular. Looking at the circuitry, we're now turning not just from RNA, but also single-cell attack, to basically look at many subtypes of excitatory and inhibitory neurons through their attack signals, looking at distinct cell type specific peaks, looking at changes that are happening late in Alzheimer's disease, specifically for excitatory and inhibitory neurons. Regulators that are able to predict perhaps interventions. So we published about SPI1 back in 2015. Now we're finding a lot of additional regulators of these immune cells that are showing the strongest enrichment for Alzheimer's disease experiments, in contrast, of course, to psychiatric disorders that are basically affecting excitatory and inhibitory neurons. Urunina bunebra, mamá. La ησυχία. Μιλάω ακόμα. Ωραία. Then predicting these regulators. And then we're able to combine both transcriptional and genomic signatures from the same cell to look at the accumulation of somatic mutations. We're now looking at these mutations that are accumulating in the mitotic cell divisions that are happening, and then how these mutations are accumulating in the case of Alzheimer's in these cells that appear to be either senescent or de-identified and losing their identity between oligodendrocytes and excitatory neurons. These are pointing to very specific pathways that are showing this mutational accumulation. And in the context of single cell attack, single cell DNA accessibility, we're finding the same signature. And in fact, a quite dramatic dysregulation of the chromatin landscape where late in Alzheimer's disease, we basically see, not in early, but late in Alzheimer's, we see this loss of accessibility for regions and this gain in accessibility of repressed regions for connectivity border. So that suggests a global erosion of the landscape. So I think I'll stop there in the interest of time. I basically talked about how we can build these reference maps and epigenomes to predict disease-relevant tissues, how we can combine genetic, epigenetic, and transcriptional variation, and how we can, in fact, carry out this epigenomic and transcriptional dissection at single cell level. Done a lot more work in dissecting the cell type, and the multi-phenotype impact of these differences, understanding how these converge across multiple tissues, and lastly, how we can in fact develop high throughput methods to understand these genomic circuitry. Popular and programmable CRISPR-Cas9 systems that allow us to impact PSCs, differentiate them into neurons, microglia, astrocytes, and oligodendrocytes, and then start asking how we can intervene at the enhancer level to activate or repress, at the SNP level, at the promoter level, at the gene level, to basically knock out or edit individual genes or nucleotides. And we've developed these very high throughput assays to basically carry out, for example, 7 million tests in a single, inserting them into self-transcribing constructs, and then using that to fine map the region the signal appears to be coming from, which basically often maps evolutionarily conserved, but also motif-containing nucleotides. And lastly, in collaboration with my former postdoc, Andreas Penning, we're now testing mutations in these microglia in the context of whole brain by inserting these high throughput reporter assays, these multiple MP arrays, into the brains of mice, and then asking about the impact of individual mutations. We were collaborating with Brad Pentelute and Marinka Zitnik to start testing many of these pathways by developing now new drugs and new therapeutics thanks to deep learning to basically start understanding how these are altered. So that's where I'll stop, to basically say that I'm very, very fortunate to be a part of this amazing team at MIT. This is from our Kellis Lab retreat. This also includes many faculty collaborators. We have both an experimental and a computational group, and we're actively looking for postdocs. Many of our former postdocs have now started their own faculty jobs at Stanford, at UCLA, at Harvard, Columbia. If any of the folks here are looking for postdocs or have students that they would like to recommend. I'll stop there and take any questions. Thank you.
Many thanks, Professor. So questions from the audience. I will have some questions, but I will wait for others. Okay, so I will ask my questions. So my first question would be rather philosophical question. So you said that many of these quantitative factors are polyfactorial. So you need to take into account many genes to actually model the output of these factors. But when you go to a single gene, can you quantify the gene expression level into dichotom values, or you need the whole spectrum of the gene expression?
So one of the things that we're looking at is basically how genetic variants contribute to dysregulation, not just at the single gene level, but at the whole pathway level. So basically, when I showed you earlier that we can start partitioning the genes into modules, we can now start looking at QTL effects at the module level. We can basically start asking about the global dysregulation of a group of genes that is pushing you towards, for example, neuroinflammation, or towards amyloid beta lack of clearance, or tau accumulation, so on and so forth, in the context of Alzheimer's, or lipid dysregulation, or cholesterol dysregulation. Basically, for every one of these complex disorders, we're now partitioning into modules and then looking at global genetic effects acting at the module level. So this is one way in which we're bridging this sort of single gene approach and the genetic impact approach at the pathway level.
But if you go to those gene factors, could you model the genes as a switch, as turn on and turn off, or you would need the whole spectrum of the gene expression?
Model them as binary. I don't think we've tried to binarize. Every single thing that we're doing is quantitative. It's basically looking at these quantitative changes. And I think that a lot of the models that we're starting to understand underlying these complex disorders is models of weak effects, models of not just shutting off or turning on, but sort of slightly altering a large number of genes and together contributing to a pathway. So I want to also mention that interventionally, we can do a lot more at the gene level than evolution tolerates. So basically, evolution at the single gene level only allows why a lot of the genetic variants are sitting in enhancer regions rather than promoters, because they're only allowed to impact the gene by so much. Protein coding and protein altering variants basically not allowed to rise at high enough frequency in evolution to be detectable as a global burden of disease. Instead, it is these common variants that are excluded from protein coding regions, that are even excluded from promoter regions, and that are even excluded from strong effect enhancer impact. Instead, it's these subtle effect variants that are only tolerated. These are acting through the accumulation, like death by 1,000 arrows kind of approach, where it's a global dysregulation that be combined with environmental insults or other kinds of perturbations that will then lead to the crossing of the threshold toward disease.
I understand. Thank you very much. My second question would be about the evolution of non-coding RNAs and the protein coding RNAs, but especially transcription factors, because many function of non-coding RNAs and transcription factors can overlap, and I would be really interested to see there is there a difference between the speed or nature of their evolution?
I mean, I'm of course fascinated by the field of non-coding RNAs, and there's been many examples where these can have dramatic effects in sort of acting either in cis or in trans. And I think more and more we're starting to understand the role of these RNAs in sort of scaffolding long-range interactions, even between chromosomes, in sort of bringing these enhanceosomes or in the sort of regulatory regions together. So in a paper that we posted in bioRxiv last year, we basically showed how these long-range contacts can happen across different chromosomes and bring together global landscapes of these modules of enhancers acting together, these very large aggregates that are happening inside the cell. You know, our current model is that RNAs are likely guiding a lot of these. And I think a major challenge in the field is sort of how to probe these effects, how to understand these condensates that are happening, how to understand the extraction from these condensates of the drivers that are leading to their establishment. For example, many of these long non-coding RNAs is actually making this dramatically harder because it's very difficult to understand these effects. But I think that's a lot of where the field is heading to now. Sort of instead of the traditional model of this turns on and that turns on and the sort of this simple circuit, we're starting to understand that it's really agglomerations and biophysical properties that are driving a lot of this. And I think RNAs are going to be very important factors.
Thank you very much. And And the third question, just out of curiosity, so you showed some really great example where one single mutation in one single enhancer can impact the phenotype significantly. My question would be that could you observe multiple enhancers with the same function associating to the same gene? You know, that there was this shadow enhancer concept back in the days. And I would be just interested because the living organism has to be robust. You know, one single so the organism can't rely on one single enhancer for a very important phenotype.
We published multiple papers on shadow enhancers with Julia Teitlinger and Alex Stark and others, you know, about a decade ago, actually. And I'm very, very interested in sort of the robustness of these biological systems. And I think, you know, a lot of those sort of dominance, recessivity effects that Mendel was, you know, talking about, we're only starting to understand sort of the extent to which this is happening in the genome and the extent to which different sort of different genes can be sort of stepping in for each other, different alleles of the same locus, different genes, different enhancers of the same gene. So I think this type of robustness is going to be found throughout biological systems at every scale, at the level of motifs in the same enhancer, of enhancers in the same gene, of genes in the same pathway, of, you know, parallel pathways. And basically, in one of the realizations that is if you test using massively parallel reporter assays multiple SNPs in the same locus, what you're often seeing is that these multiple SNPs are all pushing in the same direction, either higher or lower. If you take a purely random model of evolution, this cannot be explained. You wouldn't have a haplotype which is already lower being selected for another mutation that's pushing it further lower, another mutation that's pushing it further lower, all on the same haplotype. Basically, you know, in some cases, this may have happened when, in fact, an alternate allele that is now detrimental may have been advantageous. And I'll go back to the FTO example, which is only at 2% frequency for the rare, for the risk allele. It's only at 2% frequency in Africa, but it's at 42% frequency and 44% frequency in Europe and in Southeast Asia. So there may have actually been selection for that selection might be associated to an advantage of being able to store excess calories through, you know, periods of starvation or ice ages or, you know, long crossings through deserts and so on and so forth in the exploration of the world, in the out of Africa migrations of humans. So in those cases, you would expect selection acting on the multiple alleles pushing all in the same direction for the same haplotype. But even in a purely random model where every haplotype randomly pushing it up, some randomly pushing it down, and so on and so forth, with an ascertainment bias in genome-wide association studies, where even if this is a, you know, Brownian motion distribution, some pushing it up, some pushing it down, every now and then you would get extremes of, you know, up, 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 hitting the same haplotype. Those haplotypes are more likely to have been observed associated with disease in the context of GWAS. So you can explain it either through selection or through simply ascertainment bias. Basically, what we need to do is systematically test every SNP in the genome to start asking about the prevalence of these up-down combinations at different frequencies. And my hypothesis is that they're happening symmetrically, and it's only at the extremes that you start seeing GWAS effects.
Many thanks. And just a very last question, because I would love to hear your insight on this. You actually integrated Hi-C in your studies and show that in many cases the mutation in the insulator region can have an impact on the phenotype. So my question would be that there are many papers showing that if you repress CTCF or many other factors contributing to the three-dimensional factor, sometimes you don't really have a strong phenotype. But this is typically in a resting system, steady state system. I think that would be the holy grail of this architectural science. If you could show that what kind of change you have to do to actually see that really important impact of the three-dimensional structure on the gene expression level.
You've basically answered the question yourself. So I agree with you, basically. And I'm going to say more broadly that, yes, indeed, many of these disease-associated changes are happening in environmentally triggered conditions. So basically, I talked a lot earlier about how you basically have this accumulation of weak variants, which then compounded with an environmental stimulus will then manifest. So many of these EQTL studies have been done in control individuals. If you look at GTEx, basically our goal was to take, healthy, quote unquote, dead people. So basically people who did not die of a particular disease, but who basically died from an accident or from just an otherwise sort of healthy set of tissues. And for those individuals, many of the EQTLs that we're finding are in fact not enriched in genome-wide association studies. The challenge there might be that indeed, for most of your life, this genetic variant will not have an impact. But in the context of a disease-inducing change, then sort of having the sort of, non-risk allele, that's when it becomes important. And having the risk allele, that's when it manifests. So I agree with you completely that many of the impact of high C perturbations through CTCF or mediator or other chromatin global effect might only be visible in stress conditions. And similarly, many of the impacts of EQTLs might only be visible in the context of, for example, amyloid accumulation, old age, obesity.
Some kind of pathological condition.
Exactly.
I see. Thank you very much. I would have 10 more questions, but maybe next time or through emails I can ask them. Thank you very much, Professor, to come along and give this amazing presentation, and hope to see you next time in person.
Thank you very much, Attila, and thanks for putting together this this whole group. I think it's just so important to not only suffer from the pandemic installation, but actually to use the new technologies to bring people together across the world. So it's really a pleasure.
Thank you very much.
Across continents and to see so many people.
Thank you very much.
Thank you very much.
Thank you very much, and have a nice holiday in Greece. Bye-bye.